Auxin is indole acetic acid, a weak organic acid with a structure similar to the amino acid tryptophan. It possesses an indole ring and a carboxylic acid function. Auxin is a plant hormone that controls growth and development in different environmental conditions. Even at low concentrations, auxins can affect gene activity through specific proteins involved in signaling. Auxins are synthesized in tissues with high cell division activity like shoot tips and move through the plant using transport proteins that control their flow in and out of cells.
These are the functions of auxin, which includes vascular tissue development, root initiation, cell elongation, and regulation of gene expression.
This is the methodology section of the video. The first step is to cut two healthy stems with similar lengths from the mulberry tree. The stem chosen should be a pencil-sized stem. It is important to cut diagonally. Step 2 is cutting the stems into 4 parts, following the position of the nodes. It is crucial to leave a distance between the bottom node and the cut part of the stem because the bottom node needs to be above the soil line when it's planted. Continue to cut until there are 6 total stems, each consisting of 4 nodes. Step 3 involves dipping the cut end of the stem briefly in water to moisten it. Step 4 is preparing the soil medium in a polythene bag. Followed by step 5, adding water into the soil to moisten it and mix it thoroughly. Step 6 is dipping the stems into auxin powder until it is lightly coated. The bottom end of the stem that is wet and auxin will stick to it. Step 7 is planting the auxin-coated stem directly into the soil in the polyethylene bag. Step 8 is planting the stem without auxin in the other polyethylene bag. It is important to label both polyethylene bags clearly to avoid mix-up. Ensure the bottom node is above the soil line. The last step is leaving the plant and water daily for 3 weeks and observe the changes, all while ensure the plant has enough sunlight. After 3 weeks, we harvested the mulberry plant. Visually, it is obvious. The stems on the top are the stems coated with auxin and those at the bottom are not coated with auxin. Let's take a closer look. The stems with auxin produce larger and more leaves compared to stems without auxin that sport smaller and lesser leaves. The stems with auxin also have longer, spreading roots as compared to the stems without auxin with short or no roots at all.
After a few weeks, the mulberry plant on the left, dipped with auxin, has root growth, while the plant on the right, without any auxin, has no root growth. Other than that, the mulberry on the left is much taller and has more number of leaves compared to the plant on the right. Thus, this shows that the presence of auxin stimulates cell elongation in shoots and also helps with root formation.